So you've learned how to do Punnett squares with what's called a monohybrid cross. Now you're going to learn how to do it with a dihybrid cross. And di means two, so in this case, we're comparing organisms with two different traits. So a dihybrid cross finds the outcome of the offspring when two alleles are tested at the same time. For instance, the shape of a seed pod and the color of the peas at the same time. Now, if both parents are heterozygous for both genes, you're going to get this nice, neat ratio of 9 to 3 to 3 to 1. And I'll show you how that works in a minute. To do a dihybrid cross, the first thing we're going to do is find all the possible gametes that each parent can produce, and you're going to use the FOIL method that you would also use in algebra for expanding equations. You're going to put the gametes for one parent along the top and the other down the side, and then you're going to pair them up just like you did before. So, we talked about what a dihybrid cross is. Now we're actually going to do one. So, the example on your notes says, in pea plants, round seeds are dominant over wrinkled seeds, and yellow seeds are dominant over green ones. If both parents are heterozygous round and heterozygous yellow, what is the expected phenotypic ratio? So the first thing we have to do is figure out what the genotypes of each parent is. So here's Dad, and if both parents are heterozygous round, well, round is dominant, which means you're using an R, and heterozygous means there's one of each. So big R, little r, and heterozygous yellow. Well, yellow is dominant, so we're using the letter Y. So we have big Y, little y. And both parents have the same genotype, so they're both big R, little r, big Y, little y. And now we have to do the Punnett square. Now, what we need to do for each parent is take their genotype, and we need to figure out all of the possible gametes they could make. So remember from algebra, FOIL. First, outside, inside, last. So here's your big R, little r, big Y, little y. So you're going to take the first, and that's going to be big R, big Y. Then you're going to take the outside pair, which is big R, little y. Inside pair, little r, big Y. And last, little r, little y. And we're going to put these up along the top. So first pair, big R, big Y. Second pair, big R, little y. Third pair, little r, big Y. Fourth pair, little r, little y. Now, both parents are the same. Normally you would have to do this twice, but since both parents are the same, they're both going to have this combination. So we do the same down the side. Big R, big Y. Big R, little y. Little r, big Y. And little r, little y. And now we're pairing these up just like in a regular Punnett square, except now instead of 2 letters, we have 4 letters. So when we pair up this box with this box, we get big R, big Y, big Y. Now to do the next one down, we have big R, big Y, little y. And remember, the rule of thumb is that the capital letter always goes first. Next one down, big R, big Y. And the next one down, big R, big Y, little y. Now we do the next column, big R, big Y, little y. Big R, little y. Big R, big Y, little y. Big R, little y, little y. Third column, remember, capital letter goes first. Big R, big Y. Big R, big Y, little y. Little r, that's our first little r. big Y, little r, big Y, little y. And then last column, big R, big Y, little y, big R, little y, little y, little y, little r, big Y, little y, little r, little y, little y. Now what we need to do is take what we got and translate those into phenotypes. Well, when you think about it, there's 4 different combinations you could possibly get. Let's start with 2 dominants. You can get round and yellow. You can get round and green. You can get wrinkled and yellow. And you can get wrinkled and green. So for round and yellow, those are both dominant. So what you would look for is plants that are, they can have a big R and then anything, and then a big Y and then anything. So you would count how many have both a capital R and a capital Y. So you get 9 of those. Round and green, well, round is a big R and anything, but green is recessive, so that's going to be little y, little y. So we're looking for something with a capital R and 2 little y's. And for that you get 1, 2, 3. Wrinkled and yellow, well, wrinkled is recessive, so that's going to be little r, and then yellow is going to be big Y and anything. And for that you have 1, 2, 3. And then wrinkled and green, there's only one way because they're both recessive. It has to be little y, little y, and there's only one of those. Now if you add these up, 9 plus 3 plus 3 plus 1 makes 16, and there's 16 blocks up here, so you know you've counted them all. And this is how you get your 9 to 3 to 3 to 1 ratio in a dihybrid cross.
So we just went through a practice problem, and we saw that we got that nice, neat 9 to 3 to 3 to 1 ratio. The question is, why did we make 4 gametes for each parent? Because in the first type of Punnett squares we did, we only had 2 gametes, possible gametes for each parent. Well, the reason that this happens is because during meiosis, in metaphase 1, you have what's called independent assortment. It means the alleles line up randomly, the chromosomes line up randomly. So the alleles can pair up in any kind of manner. So what we have to do is we have to account for all of the different possible pairings. So when you're working with 2 traits, that makes 4 possible combinations. Have a great day. See you in class.